Hi dear students, welcome back to Mendelian genetics, and previously we had seen the various experiments which were being conducted by Mendel using pea plants and the various traits which he had considered in his monohybrid cross, and as well as the methods which were being adopted was also being clear to you. Now let's talk about his conclusions. The first conclusion, as we previously saw, was that the traits never blended, and this led to the formation of the principle of unit characters. He believed that the characters were not transmitted directly from generation to generation as a classical theory, but he concluded that there existed discrete particles which were responsible for the appearance of a particular trait. And he called this determining agent responsible for a particular trait as a factor or a unit, and now we call it as a gene. At his time, he never used the concept of gene, but he put forward a concept that something was responsible for every trait which was being expressed in every pea plant. And his conclusions was thus based upon the concept of unit characters, and he also emphasized that these unit characters never blended. The second principle which he came across using his study was a principle of dominance. When a pair of traits were brought together in a cross, only one trait appeared in the offspring in the F1 generation. And Mendel concluded that the trait which appeared in the F1 generation was being considered to be as dominant, and the trait which was not appearing or which was suppressed in the F1 generation was considered to be as recessive. And thus he put forward the law of dominance. And the law of dominance, it states that when a pair of contrasting traits are being brought together in a hybrid, only one of them expresses itself or dominates over the other in the F1 generation. So that was the principle of dominance which was being explained by Mendel, and it was true when he considered the pea plant as an example. In all the 7 character studies, it was found that one of each pair of the contrasting traits, it appeared to be dominant and the other was being found to be recessive. Of course, that was true in the case of Mendel's work, but later also we can find that investigators proved that dominance is not universally applicable, because there is also a phenomenon called incomplete dominance in various other organisms. So, but coming to Mendel's experiments, the principle of dominance was the one which he observed in his studies, and it was true for the case of the pea plants. Then another one that he came across was the law of segregation. Mendel assumed that every individual has a pair of factors for each character, and the two members of each pair separate when the gametes are formed, and only one pair of each pair can go to the gamete. And this was the law of segregation, or the Mendel's first law. So he was saying that even in the F1 generation, when one particular trait was being expressed, some qualities of the other parent was also remained hidden in the F1 variety. And at the time of gamete formation, what happens? The factors responsible for each trait would get separated, and in every gamete, what happens? A member of each pair is being obtained to the gamete. And that's what he mentioned, that at the time of gamete formation, the alleles or the alleles of a particular factor get segregated or separated, and it goes to different gametes. So this is the law of segregation, or it is also known as a Mendel's first law, and you can see that this law is also being considered to as a law of purity of gametes. It's mainly because He believed that each individual receives one factor from each one of its parents in respect of a particular trait, and moreover, these gametes are not found to be, or the traits coded by the gametes were not found to mix or influence the another in any way, and each gamete is therefore pure, containing only one of the factors, either dominant or recessive. And this law of segregation is also regarded as a law of purity of gametes. Now let's just explore these principles using some of the animations which are available. We had already seen the visualization of that genes do not blend, and now let's see that the genes are being dominant. Okay. Now if you cross a round variety with a yellow pea variety, in the F1 generation you get only yellow ones, and some of the second generation offsprings were being found to be green, and that is in the F2 generation you can find that there were being some of them which were being found to be green, and in the ratio of 3:1. And the results of this experiments show that the pea color is controlled by one gene which was green, which has a green form and a yellow form, and each form is a allele. Now when it appears to be as a purebred green seed plant, it has two green alleles. That is, it will have small letter y as well as small letter y, and hence it is considered to be as homozygous, as well as when it is yellow color, it has two alleles called to be capital Y and capital Y, and that's what is being depicted over here. And when a purebred green plant is crossed with a purebred yellow plant, the offspring will inherit one allele of the color gene of each parent. So this is what will happen. You have different alleles, and different offsprings will get both the alleles, and thus the F1 generation, it has been found to be heterozygous. Now here this is homozygous and this is homozygous, but in the F1 generation they will be considered to be as heterozygous in nature, because they get both the alleles from one allele, they get one allele each from either of the parent. And since all the heterozygous offsprings are yellow, then the yellow allele must be dominant over the recessive green allele, and the green allele it is not disappearing here in the F1 generation, but its effect is not seen in the heterozygotes. And the peas are yellow when they have either two yellow alleles and one yellow allele and one green allele. And the green peas must have two copies of the recessive green allele, only then it will appear to be in green in color. And now let's see how the genes get sought out, or the laws of segregation, when we cross two heterozygous plants. If you have heterozygous plants, they have what happens? Yellow capital Y and small letter y. So this is how you can see that the gametes will get separated and they will form different individuals. The ones which are having small letter y and small letter y will code for the recessive character called the green color of the seed, and the one which is having capital Y and small y will code for the what? The dominant character of yellow seeds. And in all the other heterozygous, no, the two of them are being found to be heterozygous here, and capital Y and capital Y is considered to be homozygous, yellow in color. I hope you got an idea of the laws of segregation also, which was being put forward by Mendel. With this we come to an end of the session. Thank you for now.